Chapter 26, Digestive System, Part 1, Screencast 1. Now, you'll notice that the digestive system is going to be split up into two parts. The first part will be focusing on the upper GI and an introduction to digestive function and control. And the second screencast will be limited to what we refer to as the lower GI. When we look at the digestive system, one thing to keep in mind is what its overall role is, and that is to provide a way to break down ingested nutrients. Now, this is necessary for our survival because we need to provide our body with all the building blocks that are necessary to maintain homeostasis. What we ingest is not usable in that form, so it has to be broken down, and also it has to be absorbed. Foodstuff is of no use to us unless it's actually absorbed and our body can actually do something with it. If we look at the organs that make up the GI tract, they ingest food. There's a motility function. There's also going to be lots of secretions, and last but not least, getting rid of waste products that the body did not use. As noted previously, the GI tract is broken down into the upper and lower GI, but there are also a number of accessory organs. This slide here demonstrates the difference between the GI tract and the accessory digestive organs. So note, if we look at the GI tract, we're looking at basically an opening from mouth to anus, and food actually does not enter the body until it's crossed an epithelial layer. So what we consider to be organs of the GI tract are actually part of the tube. They include the oral cavity, the pharynx, esophagus, stomach, small intestine, large intestine, and the anal canal. Whereas the accessory organs, so these are not part of the tube, but play some role with digestion in some manner. Teeth, tongue, salivary glands, liver, gallbladder, and pancreas. So probably, especially when we look at the accessory organs of the lower GI, their primary role is to produce secretions that aid in the digestion. And when we talk about the liver, the gallbladder, and the pancreas, all of their secretions basically funnel into the duodenum of the small intestine. Now, there are a total of 6 functions that we associate with the digestive system. The first, unsurprisingly, is ingestion. This is how we basically introduce food into our body so it can begin the digestion process. Motility. Now, this is going to be made up of both voluntary and involuntary muscular contractions. Also, it involves the mixing and moving materials through the GI tract. So it's not just about movement through the tract. There are going to be other movements such as churning, segmentation, etc., that we will address along the way. As noted, there are going to be lots of secretions, and so this can come from either the accessory organs or actually organs of digestion themselves. Examples of secretions will be all sorts of different digestive enzymes, acid, and also bile. Now, digestion is broken down into 2 separate processes. We look at mechanical digestion. Chewing would be an example. During this process, materials are physically broken down through chewing and mixing. Chemical digestion, on the other hand, involves the specific use of enzymes to break chemical bonds and break things down into their building blocks. So really what we're doing are catabolic processes in which we're changing large molecules into much smaller molecules so the body can actually use them. Absorption, as noted before, are nutrients that we ingest do no good until they actually are absorbed and make their way into our body, and they do that by being transported across an epithelial layer. And once that happens, they're going to move from the GI tract one of 2 ways, either in the lymph, which is specific to lipid, or our blood. The last function is going to be elimination, and we will see that our body is highly effective at getting as much as it can from what we ingest, but there will be some waste product and things that cannot be absorbed or actually digested. Now, I do want to put an emphasis on the layers of the GI tract. And we want to remember that it is a hollow tube, and it is made up of 4 layers. And those layers are from innermost to outermost: the mucosa, the submucosa, the muscularis, and the adventitia. Now, I tend to put a significant emphasis on the mucosa, and this is something that I address pretty significantly in our class meetings. Now, remember, the mucosa is the innermost of all the layers, so it is actually the layer that is in contact with whatever happens to be in the tube. So there are numerous mucosal adaptations depending upon where in the GI tract you are and what is the process of digestion that typically occurs in that digestive organ. So here, what we typically see quite often where we see lots of adaptations are the small intestine. The small intestine is the primary organ of digestion and absorption, so it is designed to increase surface area. We can see microvilli, okay? We might see lymphatic capillaries, very well vascularized areas because nutrients need to be absorbed into the blood capillaries. And also, if we look at the lymphatic system, that is how lipids and lipid-soluble vitamins will be absorbed into our body. Now, as noted previously, the mucosa is the epithelium that is in contact with lumen contents. Depending upon where we are, it can be a simple columnar epithelium, and that allows for secretion and absorption. But there are going to be some areas that are under significant mechanical stress, such as the esophagus, and they are going to be lined by a non-keratinized stratified squamous tissue. We've seen this before in areas where there is high amounts of mechanical stress. There is an underlying lamina propria, kind of think of a subfloor, and that is composed of areolar tissue, small blood vessels, and nerves. Absorption occurs when substances move into these small vessels. Now, there is a small, smooth muscle layer deep to the lamina propria, and this is known as the muscularis mucosa. Please note that this is not the muscularis. That is going to be its own separate layer. But it is smooth muscle, and contractions of this layer are going to make easier the release of secretions into the lumen, and it also is increasing the contact of material with the mucosal layer. The submucosa that is directly underneath, so once again, mucosa is innermost, then we have the submucosa, and generally we're going to see areolar and dense irregular connective tissue here, but also we're going to continue to find even more blood vessels, lymph vessels, and nerves. There is something referred to as the submucosal nerve plexus, and these are nerves and ganglia that are innervating smooth muscle and glands. We also will find in the submucosa MALT, mucosa-associated lymphatic tissue, and this is actually going to protect us from ingested microbes from crossing the GI tract. So once again, typically, whenever there's a gateway or opening to the body, we can always expect some type of immune function. Something we might see a little bit later are Peyer's patches, and those are aggregates of lymphatic nodules that we find in the ileum of the small intestine. The third layer is the muscularis, and generally it's going to be made up of two layers of smooth muscle. There is an inner circular layer, and these are smooth muscle cells that are oriented circumferentially within the GI tract, and their contraction will actually be constricting the lumen of the tube. The outer longitudinal layer, so now these muscle cells are oriented lengthwise along the GI tract, and the contraction of this layer will shorten the tube. What we will address to a significant extent is the process of motility in the GI tract. Motility is how things are moved through. It's going to be how things are mixed. And quite often, many of the diseases that are associated with the GI tract impact motility. Now, there's also a myenteric nerve plexus, and these are axons and ganglia that, between the layers, control the actual contractions of the inner circular layer and the outer longitudinal layer of smooth muscle. Now, within the muscularis, we're also going to see what we refer to as the enteric nerve system. We probably don't think digestive processes are as complicated, and I used to think of them really it's just transport across an epithelial layer. But if we think about the coordination that's necessary for the various types of motility actions that we see, it makes sense that we have to have something to control that, and that is going to be the enteric nervous system. And it is made up of two of the plexuses. Those will be the submucosal and the myenteric. And we want to think of this as local. Now, the sensory side of things will actually detect changes in the tract wall and the chemical makeup of whatever substances we actually find within the lumen. So keep in mind, because we're talking about a nervous system, there's going to be input and output. Now, along the way, sectioning off organs from each other and also closing off the lumen are what we refer to as sphincters. And they are thickenings of the muscularis layer. And they, once again, are going to be responsible for controlling the movement of materials from one section of the GI tract to the next. Now, here we basically see two types of motility that occur within the GI tract. We have a mixing. And one of the things that we'll look at it is a type of contraction that's more of a backward and forward motion. And there's no real directional movement in this type of motility. And really what it's doing is blending ingested materials with secretions. One of the examples I make of mixing pretty regularly is imagine that you're making chocolate chip cookies. And one of the things that you always do is you have to mix the chips in so that you get an equal distribution of the chocolate chips throughout the batter. If you don't do that, then there's going to be an unequal distribution of the chocolate chips. So this mixing movement is actually allowing more exposure to whatever is in the lumen, to whatever the contents are in that organ. Now, when we look at propulsion, that is a directional movement. And this is the movement used to move things through the digestive system. And this is occurring through peristalsis. And this is going to be a very controlled and coordinated contraction of the muscularis, of the GI tract wall, and think of it as a wave-like motion, because that's how this is going to be described, is a wave of contractions that are moving the contents through the GI tract. Now, the most outermost layer is the adventitia or the serosa, and depending upon where it is, it may be referred to as an adventitia. So this is areolar connective tissue, and this is found outside of the peritoneal cavity. And we have the serosa, basically made up of the same tissue as the adventitia, but now we're completely covered by the visceral peritoneum, and serosas are limited to the peritoneal cavity. Now, we've kind of talked about different types of the nervous system and actual control of the GI tract. So now let's kind of compare the two to each other. We have the enteric nervous system, and this is actually sensory and motor neurons that are found within the submucosal plexus and the myenteric plexus, and this is innervating the smooth muscle and glands of the GI tract. So primarily coordinating mixing and propulsion reflexes. So we really kind of want to think of this as a local control system that's limited to the GI tract. Now, when we look at the autonomic nervous system, this is a much more global control of digestion. And keep in mind, parasympathetic will be motor activity that promotes GI tract activity, whereas sympathetic activity will oppose activity of the digestive system. Now, if we look at nerve reflexes, so keep in mind we are looking at sensory input and then also the output. Now, when we talk about baroreceptors, they're going to be detecting stretch within the tract of the GI wall, and that's important because when there is something in the tube, it's going to stretch, and it makes sense that this will activate these stretch receptors. There also are chemoreceptors, and their role is to actually monitor the different chemical contents that we find within the lumen. Now, the reflexes, they are going to be controlled either by the autonomic nervous system or the enteric nervous system, and they are going to be initiated in response to receptor input. Short reflexes we refer to as local. They only involve the enteric nervous system, and they're coordinating small segments of the GI tract. Long reflexes, on the other hand, involve sensory input to the central nervous system and autonomic motor output. This is responsible for coordinating GI tract motility, secretions, and accessory digestive organs. Now, it's also important to keep in mind that there is hormonal control. There are a number of hormones that play a role in regulating digestive processes. Examples are going to be gastrin, which is produced, unsurprisingly, by the stomach. And then we'll have secretin, cholecystokinin, quite often abbreviated as CKK, and motilin. These are all produced by the small intestine. Remember, the exception to the rule is gastrin, which is actually produced by the stomach. Now, we next want to talk about the series of serous membranes of the abdominal cavity. I'm not going to put a significant emphasis on all of them, so I will indicate where I want you to concentrate. Now, the peritoneum, this is a serous membrane that's associated with the abdominal pelvic cavity. We have the parietal peritoneum that lines the inside surface of the abdominal wall. Then we have the visceral peritoneum, and this is a serous membrane that's reflecting over and covering the internal organ's surface. The peritoneal cavity is a space between these two layers, and keep in mind there will be a lubricating serous fluid that's originating from both the parietal layer and the visceral layer. And one of the functions of this fluid is to allow the organs found in the abdominal cavity to move freely. Now, we will describe organs based as intraperitoneal or retroperitoneal based on their position within the abdominal cavity. Organs that are completely surrounded by the visceral peritoneum, which include the stomach, the majority of the small intestine, and some components of the large intestine, are considered to be intraperitoneal organs. Retroperitoneal organs lie directly against the posterior abdominal wall, and only portions, usually the anterolateral portions, are covered with peritoneum. This would include the majority of the duodenum, the pancreas, ascending and descending colon of the large intestine, and also the rectum. We also have previously talked about the position of the kidneys, and the kidneys are very much described as being retroperitoneal as their general location within the abdominal cavity. Now, mesenteries, these are doubled layers of peritoneum, and their job is to support, suspend, and stabilize intraperitoneal GI tract organs. Within the folds, we will find blood and lymph vessels and nerves. Now, kind of imagine there has to be something that is holding our organs in position. Imagine going for a run and just feeling all of your organs sloshing around inside of you. Now, the greater omentum, this is a mesentery that covers the majority of abdominal organs, and quite often it is referred to as a fatty apron because it accumulates large amounts of adipose tissue. The lesser omentum, this actually connects the superomedial surface of the stomach and the proximal end of the duodenum to the liver. Now, there are some other structures, and I don't put as much as an emphasis on these. We have the falciform ligament. This is a crescent-shaped fold that attaches the liver to the internal surface of the anterior abdominal wall. There is the mesentery proper. This is a fan-shaped fold of peritoneum. It suspends most of the small intestine from posterior abdominal wall. And then there's the mesocolon, and this is another fold of peritoneum. It attaches part of the large intestine to the backside abdominal wall, and several distinct sections that are named for portions of the colon that are suspended.